The next section on aggregated views of proteins contains information about ligand binding sites. This section is composed out of two subsections. The first subsection contains a list of all the ligands which are directly interacting with the protein of interest. Additionally, there are ligands which can be found in PDB entries not directly interacting with the protein of interest, and you can toggle between those two modes by clicking on the tick box which is displayed. You can also use a search field in order to search for a particular ligand of interest based on the molecule name, molecule identifier, or PDB entry. And then the list just gets populated with whatever your query is. For some ligands, we do have a functional annotation, and those are sorted at the top of the list. If the annotation is known, for example, cofactor is in this case, the function is given. If you mouse over the name or the three-letter head code of ligand, you can display the name of the ligand. The link, which is directly below the name of the ligand, links to the PDBe search, which lists all the PDB entries with the protein of interest and the ligand in structure. The image serves as a link to 3D visualization, where you can inspect the ligand under question with the protein in their environment along with experimental data. In certain cases, there are multiple PDB entries which are having the same ligand bound to their structure. In those cases, the 3D display will contain a drop-down menu where you can select from different PDB entries which have the ligand bound in their structure and inspect those ligands in a greater detail. Second part of the ligand binding section contains a list of all the residues which are directly interacting with all those ligands. You can see that there is a different color coding for certain residues. This is caused by the fact that certain ligands or certain residues can be found in contact with the ligand in greater number of PDB entries. Those are highlighted with a dark blue color, whereas those which can be found in a very limited amount of PDB entries are in the light color. If you mouse over or click on any of those annotations, you can see tooltip giving you more information about particular annotation. If you collapse the ProtVista section, you can immediately see the summary for all the ligand binding sites, and immediately you can spot defined patterns in the binding. This can also help you to distinguish ligands which are of greater importance from those which may be present within that context just because of the crystallization reasons. And finally, all the data can be again downloaded either in a CSV or JSON format for your convenience.